The consolidated undrained triaxial test is a measure of soil strength with more realistic field conditions taken into account. A confining pressure is induced on the sample before the test is run, and this acts as sidewall or outer soil pressure, and in some cases hydrostatic pressures. The general concept of the test is to apply an axial pressure to the sample with simultaneous confining pressure. With the confining pressure left constant, the axial pressure is slowly increased until soil failure. For cohesive soils, a shear failure oftentimes occurs. Therefore, maximum shear strength can be calculated, and shear is the critical mode of failure. For granular soils, a bulging failure oftentimes occurs. Therefore, maximum axial strength can be calculated, and bulging is the primary mode of failure. Note that there are 4 types of triaxial tests. The reasoning for 4 different test methods is for field condition emulation. Some sites drain easily while others do not. The water table is a variable that also governs which test method is chosen. Soil conditions and overburden stresses are also major factors. The 4 test methods are described as such. First, the unconfined triaxial test, which requires just a standalone sample that is compressed. This type of test ultimately gives the user unconfined compressive soil strength. We have previously explained this test. Second, the consolidated drain triaxial test, which requires a confining stress on the sample and an open drain inlet. The sample is consolidated before compression, the drain line is left open in this case, and the sample is allowed to drain during compression. Theoretically, pore pressures can rise or fall during the test since water is allowed to drain freely into or out of the sample. Third, unconsolidated undrained triaxial test, which requires no sample consolidation and a closed drain line. The sample is not consolidated by the confining pressures, but rather left as is. The closed drain line allows pore pressures to build as the sample is compressed. Finally, the consolidated undrained triaxial test allows for consolidation to occur under a prescribed confining stress and a closed drain inlet. The sample fully consolidates under a confining stress and is then compressed with a closed drain line, which allows pore pressures to build. Today we'll be performing and reducing the data for a consolidated undrained triaxial test. Please refer to ASTM 4767 for cohesive soil preparation and specific instructions. Also reference similar standards for granular soil preparation. Ultimately, the UU, CU, and CD triaxial tests will yield Moore's circles. A unique Moore's circle will be produced for each specimen molded and tested. Note that with each new specimen molded, the molding procedure and test method must be the same throughout, except for confining pressure. The confining pressure for each specimen is unique, which will eventually produce a new circle on the shear stress versus effective stress plot. With an envelope of Moore's circles, shear strength parameters, cohesion, and angle of internal friction may be extrapolated. A consolidated undrained triaxial test is an axial compression test, but confinement is taken into account. Starting with the test name, consolidated undrained, means the sample is fully saturated and consolidated, and undrained means the sample is not allowed to drain throughout the test. To simply state the general procedure of the test, we first apply hydrostatic pressure to the sample's sidewalls through chamber pressure. Then allow for consolidation to occur under this hydrostatic pressure. We keep track of the volume change throughout consolidation. After no volume changes, consolidation is complete and the sample is ready to be compressed axially. Ensure the drain lines are closed off during compression, for pore water pressures will build during compression. Track the pore water pressure during the test along with axial stress and strain. Here is a simple depiction of the test. A confining stress or chamber pressure is added to the sample through sigma 3. An axial stress, delta sigma, is added through the bottom platen, rising as in the unconfined triaxial test, until the sample undergoes failure. The main difference between the unconfined triaxial test and the consolidated undrained triaxial test is the sidewall pressures. This helps emulate actual field conditions. Triaxial mold with a tube stem is needed. A bladder cylinder or rubber membrane fits inside the mold, holding the specimen in once the mold is removed. A vacuum must attach to the tube stem to remove air from the outer diameter of the rubber membrane and the inner diameter of the mold. A plastic pinch is attached to the tube stem to close off the vacuum when necessary. The top and bottom platens are made of stainless steel or lucite and must contain flow inlets. The base pedestal contains the valves for drainage lines and pore pressures. The black and red nozzles on the left side of the base pedestal control the left top and bottom drainage lines. Notice the top and bottom are bridged for now. We will bridge the left top and bottom drainage lines during specimen molding to keep the dimensions consistent. The connect valve in between the two pairs of nozzles controls the cell pressure or pore water pressure. We have our porous stones and filter paper, our tamper, funnel, and our test soil. Measure the distance from the base of the pedestal to the top of the bottom platen. A porous stone will sit on top of the bottom platen, so measure the thickness of the porous stone. The triaxial mold will slide down to the base as such. Now average a series of length measurements of the mold. Also average a series of diameter measurements for the mold. After subtracting the bottom platen thickness and the porous stone thickness from the length of the mold, we obtain the sample length to be 5.649 inches. The inner diameter of the mold is 2.992 inches, therefore the area of the sample is 7.030 square inches, and the volume of the sample comes to 39.721 cubic inches. After placing a porous stone and filter paper onto the bottom platen of the pedestal, slide the bottom end of the membrane off the mold and onto the platen. Turn the vacuum on to remove any air between the membrane and the mold. Ensure that the membrane covers the whole diameter of the bottom platen. Push the O-ring down into the groove so that it seats itself and holds the bottom of the membrane in place. This way the soil will not come out from within the membrane during molding. Scoop or spoon soil into the funnel. Add enough soil to fill approximately 1/6 of the exposed length. Tamp soil to the desired degree of compaction by dropping the tamp at equal heights. Record the number of drops per lift. Place and compact the remaining 5 lifts in the same manner as the first. After the last lift, place a filter paper on top of the soil, then pour a stone and top platen. Slide the top end of the membrane off the mold and onto the top platen. Once the membrane fully covers the top platen, bridge the top and bottom nozzles with tubing. Connect the vacuum to the bridge line so that the sample will remain upright after removing the mold. Carefully remove the mold. Slide the O-ring down onto the top platen to seal off the top. Connect the drain lines to the top platen by pushing or screwing them into the inlets. Clean the base pedestal and wipe away any soil that may have spilled during molding. After the base is completely clean, grease the bottom gasket on the pedestal. Grease the bottom of the cell. Place the cell over the specimen and slightly turn it to the right to seat it within the gasket. Grease the loading cap gasket and the top of the cell. Place the loading cap onto the top of the cell body. Turn the cap so that its rods align with their respective positions on the bottom of the pedestal. Insert the rods and tighten the wing nuts down, ensuring the cell becomes watertight. Tighten the wing nuts evenly to seat it flush onto the cell body. Connect the fill valve with the fill hose. Fill the chamber completely with de-aered water. An air release should be open at the top of the chamber to ensure the chamber is completely filled with water. Once water begins to come out of the release, close the air release valve, turn off the fill water and the vacuum. Connect a drain line from the board to the red nozzle inlet so that the regulator controls the red or bottom inlet. Similarly, connect a drain line from the board to the black inlet so that the regulator controls the black or top inlet. In our case, regulator 1 represents the top and regulator 2 represents the bottom. Regulator 1 is switched to vent mode so that the top inlet may be open to flow. Regulator 2 is switched to pressure mode so that the flow will be forced from the bottom of the sample. This will cause water to flow up through the sample, fully saturating it. Increase the hydraulic head in regulator 2 so that the water level in the manometer is above that of regulator 1. Allow draining to occur. Notice the hydraulic head is decreasing in regulator 2 and increasing in regulator 1. Thus, water is flowing down from the manometer and into the bottom platen, through the specimen, and up into the opposing manometer. Allow for the water levels in the two manometers to equilibrate, then repeat the process to ensure full saturation. Fully saturating the sample may take hours or even days depending upon sample type. Once the saturation period has ended, we will next apply a confining stress to the cell body. Plug the hose from regulator 2, pressure regulator, into the main inlet between the pairs of nozzles as shown. Then turn the master regulator knob to the desired confining stress. As shown, the cell pressure is increased to 1.5 psi. Record the water level in the manometer that is defined by the top of the sample. We will be tracking the total volume change during consolidation. In our case, the water level in regulator 1 is read to be 14.5 before applying the consolidation pressure. Turn the bottom switch on regulator 2 to the on position. The confining stress is now applied to the specimen, and the volume change is tracked through regulator 1. The water level slightly rises in regulator 1, indicating void expulsion through confining stress. Allow for the hydraulic head to equilibrate in regulator 1. Once the head is stable, the sample has finished consolidating under that confining stress. Notice that the water level in regulator 1 has stabilized at 13.2. The volume difference between water level readings 14.5 and 13.2 will now have to be converted into some measurable unit of volume. The volume change experienced throughout consolidation was 0.70 milliliters, which is 0.70 cubic centimeters. Converting to units of cubic inches, the volume change becomes 0.0427 cubic inches. Close off the red nozzle to ensure drainage does not occur during loading. Remove the black nozzle hose from the board and attach it to a pore pressure transducer. This will allow pore pressure to be read out as a sample is compressed. We expect pore water pressures to increase as the sample is crushed, for the test does not allow drainage to occur. Note that the black nozzle should be in the open position at this point. Keeping the confining pressure on the specimen, move the triaxial permeameter to the load cell. Attach the bushing to the load transducer so the top platen is in series with the bushing. Position an LVDT so that it is capable of measuring upward movement by the master loader platen. Set the loading rate to 0.5% strain per minute. Tear the transducer and LVDT to 0. Prepare to record load, axial deformation, pore water pressure as the master loader moves the specimen upward. Push the up arrow to start the test and begin recording in an incremental fashion. Run the test until failure or 15% strain occurs. Failure is normally characterized by sample bulging or natural shear failure. In our case, notice how the bottom of the sample bulges. Our sample undergoes a bulging failure, which is characteristic of sand. The table displays the results to the consolidated undrained triaxial test for the first run of a full test, defined by a confining stress of 203.4 kPa. The first column is the axial stress of the specimen, load, P, divided by the corrected or actual area. The load is directly read off from the transducer, and the corrected area is the original area of the specimen divided by 1 minus the axial strain, where the axial strain is the displacement divided by the original specimen length. Note that the displacement is read off from the LVDT. Pore pressure is the second column, and it is read directly from the pore pressure transducer, which was attached to the top outlet hosing throughout the duration of the test. Pore pressures build in the line as the sample is compressed. Because water is incompressible and the sample is not allowed to drain, pressure builds up as the platen rises. The third column is effective stress, which is the stress on the sample if one were to subtract the pore influence, thus only the axial influence from the moving platen. We expect the effective stress to increase as the platen rises. The fourth column is shear stress, which is the confining stress subtracted from the axial stress on the specimen divided by 2. The following columns are variables that have been previously explained. To complete a full triaxial test, 2 to 3 separate tables such as this will need to be prepared. Therefore, mold another specimen the exact same way we did the first, then apply a higher confining stress than 203.4 kPa and compress the specimen. For instance, our next confining stress, we use 410.2 kPa. Here is a depiction of Moore's circles. The x-axis is effective stress and the y-axis is shear stress, both in units of kPa. Each of the two circles is defined by a different confining stress. For example, the smaller of the two circles displays results from a confining stress of 203.4 kPa, and the larger displays results from a confining stress of 410.2 kPa, thus representing the first and second runs of our test. So 2 separate tests were run in order to achieve the 2 circles. To make this construction, we will look at the 410.2 kPa confining stress circle as an example. We first plot the point defined by the effective stress and shear stress at failure, which is the green arrow indicator. Next, select the largest effective stress point, maximum principal stress, which in this case was approximately 965 kPa. Notice how the far right-hand side of the circle intersects the x-axis at this point. The next selected point is the minimum principal stress, which is 200 kPa. If we connect these 3 points, a circle should form with the center being about the average of the minimum and maximum principal stresses. Next, the line tangent to both circles is drawn. The slope of this line is defined by phi, the angle of internal friction, unique to the soil. The intersection of this line with the y-axis defines the cohesion of the soil. A sand displays no cohesion, so it makes sense for the line to run through the point 0, 0 on this plot for our soil. The series of circles for a test soil drawn in this manner is known as the failure envelope. Any shear or effective stress point that plots above the tangent line defined by the envelope represents failure. A plot of shear stress as a function of axial strain is shown. The higher confining stress, the higher shear capability the sample has. For the 410.2 kPa confining stress displayed an ultimate shear capacity of 375 kPa, and the 203.4 kPa confining stress displayed an ultimate shear capacity of 100 kPa. Each of the two plots displays a linear portion, peak at failure, and residual straight line portion post-failure, which is similar to the results of the unconfined compression test. Other popular plots for the consolidated undrained triaxial data include average stresses versus strain, void ratio versus strain, and pore pressures versus strain. The final plot is a graph of pore pressure change as a function of axial strain. We expect an increase in pore pressure with increasing axial strain, for the sample is being crushed without draining occurring, so water pressure will build due to its incompressible nature. Notice that the pore pressure changes do not occur until a later axial strain, about 1.5%. Until the specimen reaches 1.5% strain, pore pressures have not changed much at all. After 1.5%, an almost linear data set is realized until failure of the specimen, which occurs between 35 and 40 kPa. This concludes the data reduction and procedures portion of the consolidated undrained triaxial test laboratory.